If you are preparing for JAMB, listen to me carefully. I'm going to reveal something very, very important to you. But before then, take a look at these questions. These questions were thrown from a very critical topic in biology, and that's heredity and variation. Now, first, what I observed is that of all the topics in biology, JAMB asks more questions on this particular topic. I have observed over the years that some years JAMB asks as many as 6, 7 questions from this topic alone. That is very important. And many students out there they don't know which topic many questions come out from. They don't know which questions are more likely to be repeated, and so on. So this is the number one thing I want you to understand. I'm going to still tell you another secret that a lot of people out there don't know. If you know the topic that JAMB asks a lot of questions, you should be able to understand that you are expected to prioritize such topics. In other topics, JAMB asks 1 question, 2 questions, 3 in some topics, and some topics they don't even ask some years. But many out there don't know all these things. So they just be reading continually. They don't even know which area, as in they don't even care. But let's even do this. Very quickly, look at this question. Evaluate yourself on these questions. Be very practical and also honest. Will you be able to answer these questions confidently, as in you are sure that this answer that I have chosen is correct? Because over the time I observed that questions like this are being repeated over the years. For example, I just decided to pick these questions from 2013 and 2016, two questions. I have seen in some of the years that JAMB asks even up to 7. Don't worry, I'm going to do a video where I will show yes and the number of questions that JAMB asked under this particular topic. I've already set the questions. I've already extracted the questions from JAMB past questions, and very soon I'm going to do another video. So because of the criticality of this topic, I decided to look into the syllabus and see what learning objectives does JAMB expect from you. And this leads me to the second secret. The second secret is that if you know the objectives, the learning objectives JAMB wants to achieve in each topic, then you don't have anything to panic about. What this means is that the objectives under each topic are the questions you should expect. And imagine you know the questions you should expect in exams. That looks like expo. Many of you don't know. Okay, let me say this very quickly. Why am I even talking like this? I've scored about 338 in the JAMB equivalent exam that I've written that gained me entry into the university during my days. And by the grace of God, I'm a first-class graduate. I've adopted various learning methods, various learning techniques to be fast, to be smart, and to be accurate. So that is why I'm telling you this. So once you know the JAMB syllable objectives, I'm telling you, you can predict their question. All you need to do, know what they expect you to know under that objective. Let me quickly go into this because we are going to look at this topic critically. I'm going to explain some key things based on what JAMB has said in the syllabus. And at the end, you'll be able to answer questions like this easily. All right, let's continue. Okay, so this is a build-up of the previous video I have made. If you are my student, you know the last assignment I gave you that you submitted to me via WhatsApp in our group. I covered continuous and discontinuous variation. I covered a lot of things still under variation. So this is, and I told you that is part 1 because it's a bit longer. That's why I just decided to make it into 2 videos. So if you are my student, this is just a continuation. If you are not my student or you are just coming across this video, know that there is another part that you are supposed to watch. Just check on this channel, a video on variation. I think it should be, yes, just below this video. If you go to the channel, you would see it. So, we have covered objective 1 to 6. Now, this is objective 7. Objective 7 says that candidates should be able to identify physiological variations among human population. What does that mean? If I say you should be able to, you know the rest now. You know the rest. You are expected to know. So, which means you should be ready for question in that area. So, I've talked about morphological variation. And by the way, I don't want you to be confused. Remember, when we're talking about types of variation, we talk about continuous and discontinuous. Now again, these morphological and physiological variations are also types of variations. Meanwhile, continuous and discontinuous are a broader classification of variation, unlike this physiological and morphological, and you are supposed to know the difference. Very important. So under morphological, we talk about the physical and the internal differences in organisms. But when we are talking about physiological, we are talking about physical and the process inclusive. For example, take a look at this picture. You will see people with different skin color. Yes, of course, they are physical, which also is part of morphological. Yet, this skin color are also controlled by genes, by the DNA, by the genetic material, by alleles. So that's why it has to be under physiological variation. I don't want to waste much time. Please watch the previous video. You will get it very well. So let's move further. So this is just a recap of morphological and physiological variation. I'm not going to waste time here. Please just permit me. I want this video to be as short as possible. So by the time I share these slides, you'll be able to go through it again and get what is there. Meanwhile, if you are not my student, just drop in a comment section that you want to get the slide. I will share it with you. So morphological, we have talked about them. In general, or in summary, there are differences in the physical characteristics, either external or internal. We have height, weight, skin color, and so on. While when you talk about physiological, the first thing that should come to your mind should be the living system function or the process or how it operates. This talks about blood group. For example, blood group is a discontinuous variation. Of course, you know, we dealt with that previously or in the previous video. You know that blood group is a discontinuous variation because it's not continuous. You can either be AB, B, or O, no other group. But when we talk about height, we can find someone maybe 5.5 meter, 5.3, 5.2, 5.1, 5.0, 5. You see that is continuous. So, but in blood group, for example, in discontinuous, you can either be here or there. No two ways about that. Please permit me to move on, I beg. I don't have much time. All right, so JAMB also said that you should be able to know how blood group is important in blood transfusion and paternity determination. Remember, this is a continuous variation. So we are now talking about the application of continuous variation. And these are some of the questions that are very interesting when you are asking questions, because it is not theory. There should be practical application of it. So JAMB said you are supposed to know the application of this discontinuous variation, which has to do with blood group. So you have to know its role in blood transfusion and in paternity determination. I'm not going to talk much here, but let me give a very brief explanation of this table. Please let me beg you, because there is no year they don't ask question under this blood group around this blood group matter. Yes, crime detection, forensic, and so on. Please let me beg you now, master this table. By the time I send this slide, these are some of the key areas you should give more attention to. Okay, so we have ABO blood group system. So we have blood group A, we have blood group B, we have blood group AB, and we have blood group O. Now, a person with blood group A, listen carefully and understand. It's very simple. Because I'm going to explain antigen and antibody now, which are very important. A person with blood group A has A antigen. What is antigen? Antigen is just a part, usually maybe a carbohydrate, a protein, or something that can be detected by an antibody. I believe you know what an antibody is. They are proteins that fight against foreign substances. They are part of our immune system. That is to say, if you have any microbe, if anything enters into your body, those soldiers are there. So how do they detect that this is a foreign organism? This is not part of us. This is not part of the body. Let's destroy it. They detect that by that antigen. So a person that has blood group A has a surface antigen A. He has a surface antigen A. So such a person will have antibodies B. Because if he has antibodies A, those antibodies will recognize the blood cells of that person. Since he has antigen A, he can never have antibody A, because antigen A is captured by antibody A and is destroyed. Antibody A will recognize antigen A as a foreign invader and it will destroy it. I hope my explanation is very simple. Please, the comment section is very important. That's why it is there for you. If not, if something is not clear, let me know. So, if you have blood group A, definitely you cannot have antibodies against A. If you have antibodies against A, there is a problem already, because those antibodies will invade your red blood cells and they will destroy them, because they are detecting that antigen A as a foreign thing. So instead of your body to see your blood as part of it, it will see it as not part of it and it will destroy it. I'm going to make this more clearer. If you have understood this, I don't need to explain B, AB, and O again, because that's just the basics. It's what applies to others. It means that if you have blood group B, you have antigen B. So you have antibody A. So antibody A will not recognize antigen B as an enemy. It will only recognize antigen A as a foreigner or as a foreign invader. That is why if they mistakenly give a person with blood group A the blood of a person with blood group B, there will be reaction. We call it agglutination. The blood will clot together. The antibody, because look at it now, look at what is happening. The antigen A, I am antigen A, I have blood group A, antigen A, and I have antibodies against B. My body has antibodies. It has soldiers that kills B. And you are now giving me the blood of somebody with group B. Immediately my immune system will sense that this is a foreigner. Let's destroy him. So all my antibodies against B will capture those red blood cells and destroy them. So this is what happened. This is blood matching. You have to just get it right. You just have to get it right. I remember when I was on Syllabus, there was such a kind of, should I call it an error or a mistake? Okay, so a patient was given a wrong blood group. Maybe at the bank, there's what we call blood banking. So maybe at the bank, they mistakenly, or the person that labeled the blood, label it as A instead of B. So immediately they start doing the transfusion, putting the blood. The person started reacting, the body started shaking everywhere. So they quickly have to disconnect it. That's just it. I'm not going to explain the other part like I said. So let's continue. You should know that blood group A can collect from A, can collect from O. Blood group B can collect from B, can collect from O. Blood group AB can collect from all. We call him universal recipient. However, It's not every blood group ABO because we also have rhesus factor. Blood group A positive, we have blood group A negative, rhesus factor negative. That negative can only receive from blood group AB negative. But generally, we say blood group AB is a general recipient, while blood group O is a general donor. Please, I'm not going to explain these things more. By the time I send this thing, you should be able to go through it. We need to be very fast. Okay, so I don't need to talk about this here. Remember, we're talking about the application of these things. So by the time I send, please, you can read all these things. It's just about transfusion. Then in paternity, yes, this is very important. Let me make it clear. Someone once say, my dad is blood group O, my mom is blood group AB. No. My dad is blood group O, my mom is blood group O too, but me, I'm blood group A. I laughed. I said, please go and do check up again. You know why? Okay. So it helps in determining paternity. If your dad is blood group O, your mom is blood group O, there is no way you can get blood group A or blood group B or blood group AB if you are a child, the child of your mother. I didn't want to cause trouble with that other friend that told me that his mother and father are O and he is A. No, I don't want to. Let me not say, do you not know? No. All right, so that's it. If your parents are OO, for example, a child would have to be O, is compulsory. Now, if the father is A and the mother is O, A always dominates. So all children are not even likely, all children are going to be A. So A dominates O always. B also dominates O. So parents with blood group AA, child will be A. Parents with blood group BB, child will be B. But if one is O, another is B, all the children will be BB because the A and B always dominate. A and B always dominate O. So for a child to be blood group O, it means that all the parents are blood group O. I hope this is very clear. You may see this in exam because it's real life applied question. And that's how they use it to determine who is the father. It does not tell accurately, but it's a basic test that will help you. For example, someone say, it's not my child, and they call someone and say, this is the person's child. All they need to do, what's the mother's blood group? What's that suspect's blood group? So if the mother is O, the suspect is O, and the baby is A, that man cannot be the father of that child. Because where did the child get A? When the suspect is O and the mother is O, it's not possible. So it gives the basics. Okay? So let's quickly move. I don't have time. I want to be very fast. So the JAMB also said that you should know how to apply discontinuous variation in crime detection. I have already given one example. In crime detection, blood group, it can help, it can give the basics, but not complete. So fingerprint is a discontinuous variation. Because we don't have in between. You either fall under whole, loop, arc, and whatever. In the previous video, I have covered this clearly with clear explanation. So in crime detection, we have what we call DNA profiling. All this, if they have a dispute of a child, they will go and do DNA test. So that one is also a discontinuous variation. Your DNA is completely different from any other person's DNA, but it can be close to your parents. So if there is a dispute in a child, they will go and do DNA test. That will give. So that DNA is a discontinuous because you cannot have in between. You will have your own specific. There is no close to, and so on and so forth. So I hope this is clear too. Okay, now very quickly. JAMB also said you should know how to determine heritable and non-heritable characters. I think this is very simple. How do you do it? There are 2 ways, or 3, or even up to 4, I think, yes, that you can do to know. For example, in this image, first generation, we've seen a specific trait. Second generation, maybe is missing, and third generation, it shows up. So it's possible the baby has copied it from the mother or the father. So how do you know? By doing what we call pedigree. You follow the family tree and check. Oh, so person. Oh, the grandfather was so that is another way is to study twins, identical twins, monozygote twins. If you study monozygote twins, it will give you a picture of heritable characters. So heritable characters are passed from parents to offspring, like eye color, blood group, earlobes, and so on. Non-heritable, they are not passed through, like tattoos, scars, muscle build in gym, and so on. So you can determine by twin study, pedigree, and so on. Please, you can go through all this. Yes, JAMB say you should know the basics about DNA. And I saw questions under this DNA. Please, I beg you, know the salient features of DNA. One, you should know that it's double helical strand or double helix structure, whichever way you want to know. No, you should know that it is made up of base pairs. Base pairs. I'm going to show it in the next slide. And the base pairs are joined together by hydrogen bond. This may not be too important for now, but just know that it's a double helical strand. So let's look at this now. You should know I saw a question under this. And yes, one of the questions I showed in the first slide is also among these questions, because they are saying, how is the pairing done? Please, this is a rule. By the time you advance, you will know all this Shagaf's rule, how the base pairing occurs, and other salient features. But for now, please know, we have base pairs of DNA. We have guanine, cytosine, then we have adenosine, and thiamine. So these four, how do they pair? Thiamine always pairs with adenosine. So T and A, or A and T, TA or AT is compulsory. A cannot pair with cytosine with C. A cannot pair with G, but A can pair with C, and with T, T can pair with A. These questions are real, and I see them, a lot of them in this area. You have to know. They will either ask you to show which pairs with which, or they will ask you which one has double bond or triple bond, or they would ask you the names of this bases. So G and C pairs, A and T, or T and A, G and C, or C and G, you can swap them. Anyone can come first. So I'm saying this so that you know when you see the right answer. So the way I mastered it when I was in the university was AT. I use an acronym at GC. That's just what I did. At GC. So any day you wake me up and say, how is the pairing done? I'll just remember at, which means AT, GC. So A and T, G and C, or T and A, C and G. Anyhow you swap them, but it must be A and T, or T and A. So know that base pairing. A and T always have double bond, and C and G always have triple bond. Please, I beg you know this. I've mentioned three down there: adenosine, thiamine, guanine, and cytosine. Okay, I can go. Functions. What's the function of DNA? It carries the genetic information. Yes, JAMB talks about segregation of genes in meiosis and recombination at fertilization. This is a bit technical, but I will try my best to explain to your own understanding. So first of all, there is what we call DNA. I've already explained the basics of DNA. This DNA is what carries genetic information about offspring, and each character is coded by a gene. For example, skin color. There might be genes that are responsible for skin color. That's why okay, let me leave that for later. I will explain it. So during meiosis, you remember meiosis and mitosis. We have somatic cell division. In somatic cell division, we have mitosis. That's where cell divides in number. But in the type of cell division that occurs during reproduction to form gametes, to form the sperm and the ovum, that one is different from mitosis. It's not just direct separation to have two daughter cells. No, it doesn't happen like that. So half of the information are transferred, and each trait has two genes, dominant and recessive. By the time I explain, maybe I go further with explanation, it might be clear. Let me give an example. A father is a very tall man and the mother is a very short woman. Now, the character of tallness is in the woman, even though she is short. But that character of tallness is not dominant. It has not shown, but is there. So you see that it's two now. She has two, tall and short. Even though she is not tall, she is short, but the tall is still there. Short is dominant, tall is recessive. It did not show. The same thing with the father. A tall father has the gene, the character of shortness in him. That's the fact. That's why if the father is tall, the mother is tall, they can still give birth to a very short person because they have the... Now, during gamete formation, these two characters are separated now. If the mother is short and the father is tall, now let me tell you what happens. During meiosis, there will be a transfer. The two will separate. So we have the trait, the characteristic for shortness, which is showing in the woman, and the characteristic of tallness, which is not showing in the woman. Now, each of these two will separate, and one will enter into one egg, another one will enter into one egg. I hope this is clear. The same thing with the father, the sperm. During the sperm formation, there will also be a segregation. There will be a separation. One will enter, of the tallness will enter one, and of the shortness will enter another sperm. Now, listen carefully. What will be the end result now? Depending on which gamete of the father fertilizes which gamete of the mother, that will determine what we will see as an end product. For example, the woman has tallness as recessive, not showing, but is there. If that egg is fertilized by a sperm that is tall, which is showing in the father, the baby will become tall. If the dominant character of the mother, the recessive rather, the short, fertilizes with the short of the father, you know the end of the story. Please, I think these are basic things in biology. You remember crossing? Yes. Please. If it's not clear, let me know. I don't want to waste much time. So this is the whole thing that this image is trying to tell you. I just use my own explanation. So you have to know what segregation is, fertilization. I've explained all this. This is still talking about segregation and fertilization. It's showing how the genes are separated during the formation of gametes. And this is guided by the law of segregation. Good. So I've explained all this. Now look at these two birds. The purple and the pink. Is this pink? Okay, so the two came together. The one that is dominant is what will show eventually in the baby. The recessive is still there, or the baby will be the carrier, but it will not show. Maybe it might show in the next generation. Okay, so you should be able to deduce recombination and also segregation. I won't talk much here. By the time I share slides, you will use it. So you should be able to analyze data. Hey, you should be able to do crossing. You should be able to do statistics, probability. What's the probability? The father is this, the mother is this. What is the probability that their child is short? Is going to be short? Is it 2/4? 4, that's 1/2, 50/50, or is 1 out of 4, and so on and so forth. I think these things are very simple. Do you really understand them? Please, because I don't want this video to be long, if you want me to do a separate video explaining this crossbreeding and whatever, please just let me know in the comment section. I will do that. For now, I don't want to really take much time on that because that's not the priority. Meanwhile, JAMB say you should know it too, so which means you should expect questions. In fact, one of the questions I also showed there was asking about reprobability, whether it's 50%, the father is carrier, mother is this, and those are applied questions. So all you need to know is be able to do all this, the Punnett square analysis, and be able to tell the end result. So, if the father is tall, the mother is tall, do I need to explain all these things, for God's sake? Okay, if the father is tall, the mother is tall, or look at this image. Let's use the image to explain quickly. SS, capital letter S, let's say is tall. Okay, small letter S, let's say is short, the mother. Then the father will give one of the alleles, which is for tallness. The mother will give one of the alleles, which is for shortness. So their offspring will be tall, but carrier of shortness. So in the next generation, we would have shortness as dominant. We'll have shortness as dominant in one of the babies. So usually you would only need to master the first generation, first filial generation, and with it you are good to go. If you have 2 alleles of dominant and recessive character, please let me go. Crossbreeding. So application of hereditary principles, very important. JAMB say you should know it too. Is their objective 6 under heredity. So what's the application in crossbreeding? You can select parents of a desired characters and crossbreed so that you can get what you want. You can produce hybrid of improved crop varieties. What is the technique? Selective breeding and hybridization. Yeah. Okay, I'm going to explain something very, very critical here. Listen to me carefully. There's what we call outbreeding and there's what we call inbreeding. JAMB says you should know them and their advantages. So when we talk about outbreeding, it means you are breeding an organism with another organism that is not closely related to it. Okay, let me give an example. In marriage, typically, what we do is outbreeding. You don't marry someone that you are closely related to. Yes, it's very dangerous biologically. Some of you don't know. You don't know that if a brother marry his sister, they will give birth to very weak children, unfit. They may not even be able to survive. Are you hearing this stuff for the first time? So that's biology for you, and it's real. It's real. In humans, this happens. So God is wonderful. That's why He said there should be no incest. A brother with sister, no, now it does not make sense. So if such people happen to even come together, their children will be very weak. They might die fast. They may come up with different kind of diseases. That's why we don't marry close blood people. So that's what we do actually, outbreeding, marrying someone from far distant, from that is not closely related by blood. So what is the advantage of that? The advantage is that we can have different combination of characters in the children or in the offspring or in the end product, which increases diversity. Is an advantage. But what is the disadvantage? The disadvantage is that the pure trait, the pure characters of that family will be lost. Because by the time you give birth to your child, of course, because the father comes from another place or the mother comes from another place, you cannot have a direct similar characters of that of your family. You may have in some cases if your maybe as a father or the mother have dominant characters in the child, so the resemblance may be much, so and so on. In the case of inbreeding, that's closely mating or reproduction by closely related organisms. So what's the advantage? It maintains desirable character like homozygosity. The characters would continue the same. But what's the disadvantage? Reduced fitness. This is what I was talking about. The children will not be fit and they will have genetic disorders. They will come up with all manner of genetic disorders. So what are the applications? In animal breeding, for example, if you see a cattle that has a particular body size and you desire it, you can cross it with another cow for mating. So they will produce a better breed. Then in another place, in inbreeding, if they want to have a particular kind of dog, maybe dog that has plenty for, a particular shape. So instead of them to allow that dog to mate with another, they will allow it to mate with only its own kind. So to maintain that kind of dog nature. If I share, you will know, please. You will get some other things. I may not be able to share. I don't want the video to be long. So we have GMOs. I am sure this is not your first time. Many of us have been consuming these genetically modified plants. The most common ones, the popular ones are maize, soybeans, papaya, rice, and so on and so forth. Tomato, potato, they are all there out there. So, but JAMB wants you to know the contentious issues. You see, I tell you that these things are very, very simple. Just know the objective and you will know the question to expect. So are there contentious issues? Yes, there is contention about GMOs. Let's look at the next slide and see. I'm almost done. So they are genetically modified organisms, of course, and they come with advantage. They can be pest resistant, disease resistant, drought resistant, for example, in the case of plants. But what are the pros? The pros is we can have high yield crops if we do crossbreeding of plant genetic modification of organisms. We can have cattle that can produce plenty milk than usual ones. We have poultry, fowls now. You know that they are not like our normal traditional fowls. They are genetically modified. So they can grow fast. They can have a lot of meat, far more than the normal. So those are the advantages. We can have high yield of those organisms. What's the problem? Environmental impact and health concerns. Over the time, people are saying, are consuming these things really healthy? So there is a concern. In fact, some people don't allow it. Some countries don't allow GMOs. So it's a contention. People are contending. We should not do it. We should not do it. While some are saying, we can get more yield. We can get a lot of food for people and so on and so forth. Then we have gene therapy. Gene therapy is treating disorders by inserting gene like cystic fibrosis. If a gene is faulty, I said a gene controls a particular character. Let me give you an example. Have you ever seen poultry with different color of feather? Yes, there is a gene that is responsible for control of that feather. So if you remove the cell of that kind of organism and remove that exact gene, trace the gene that is responsible for the feather color and remove it and replace it with the one that will produce red color, that fowl will continue to produce red feathered children. This is just it. It's very simple. So it's the same thing. If a particular gene has been discovered, like in this cystic fibrosis, there is a gene that is faulty, so they can go and trace it in the DNA, cut it out, replace it with a healthy one, and the person becomes healed. That's what we call gene therapy. So it is used to cure diseases, but there are still ethical issues, there are still challenges, there are still contentions. People are still saying, what are the side effects? The ethical issues and so on and so forth. So they are afraid that one day scientists will create an organism that will be out of control. So I think maybe I've had something like that. Let's continue, please. Applying heredity knowledge in marriage counseling. If you have watched to this end, you are doing yourself good. You are doing yourself good. I'm almost rounding up now. I think second to the last or so. Yes, so In marriage, heredity is also applied. JAMB wants you to know the application of heredity in marriage counseling. Very important. So, focus is blood grouping. Your blood group may not really matter much, but it matters. Then we have sickle cell anemia. Yes, they will say go and do genotype before marriage, which is very important. Yes, so that you will know your sickle status, sickle cell, whether you and your intending husband or wife are good to go. Okay, then we have resource factor, which is really talked about and is very important. So, how is it applied in marriage counseling? If someone is SS and the intending husband or wife is also SS, you see, you can use heredity for counseling to say, we advise as medical practitioners. Some of you are going to be nurses, some of you are going to be medical doctors one day. So you are going to meet this. Sure. I hope you will remember to call Mr. Emmanuel and say, sir, I'm meeting what you have taught me. All right, so that's that. So it helps to do screening. So this resource factor, I said is less talked about, is very important. If the mother is resource factor negative and the fetus is resource factor positive, there is a risk for hemolytic disease, though it can be prevented by injection if known before delivery. Very important. So this is the application of heredity in marriage counseling. Yes. What's the significance of recombinant DNA? JAMB wants you to know this, though this is a little bit advanced, but you are expected to know it anyway. Now, there are some drugs that are produced using recombinant DNA technology. I will tell you what recombinant DNA technology means. I told you that each gene is a portion of DNA that controls a particular trait or that produces a particular protein or whatever you may call it. Now, there is a particular drug called ampicillin. Ampicillin is actually produced by microorganisms. So microorganisms are known to produce it. Why do they produce it? They produce it as a defense so that wherever they are, other microbes cannot exist. So that ampicillin is able to kill microorganisms. It's just a normal defense system. So scientists will now get the microbe that produce ampicillin and grow them. They will put food for them. We call it agar or culture rather. And those microorganisms will grow and produce a lot of that substance. Then they will kill all the microorganisms and they will extract that particular compounds and they sell it as drugs. So that's I'm going this way because I want you to get the real thing. So now when we come to the recombinant DNA technology, some people have this issue of diabetes. We have types of diabetes. Some people cannot produce insulin, so they have to give them insulin injection. Some of you might have heard that. If you have not heard that, please go and research SS3, SS2. You should have heard of this. Okay, so now what do? The person cannot produce insulin, and insulin is needed. So you cannot kill another person to get insulin to inject the other person. That will make sense now. So before, they were using bovine insulin. Cows produce insulin. So it's still under genetic stuff. So they will use bovine insulin and be injecting humans. But later on, they discovered the gene, the portion of the DNA that leads to production of insulin. So they cut it out through the recombinant DNA technology. They put it inside a bacteria and then they grow that bacteria. So all of a sudden, that bacteria begin to produce excess of insulin. And they will kill all the bacteria, extract the insulin, and package it in form of drugs. So this is the application of recombinant DNA in medical products. It's as simple as that. This technique I just told you is applied in various areas, like in food industries, in whatever. There are a lot of industries. So, but I hope this is clear. This image just show you the fermentation tank is where the microbes are grown under a particular temperature. Food is given to them. They will grow fast. Microbes grow as fast. Some they can grow 1,000,000 times in few seconds or minutes. Depends. So they grow fast and produce what is desired. They are harvested, killed, and then extracted. The desired product is extracted. So that's that about by the time I share this, please you can read this. Yes, sex-linked characters. Permit me to check. Okay. Oh, we have only 2 slides to go. So sex-linked characters. There are characters that are connected to sex. Some are not connected to sex. So sex-linked characters are characters that are linked to sex. Like I said earlier, mostly they are X-linked. We know X is from a woman and Y is from the mother and the father. So examples are hemophilia, color blindness. Why does genetics diseases come from a mother? Why? The reason is because if, for example, that's why male usually suffer more from them. If a girl is giving birth to, and maybe the father has a genetic disease, because he will donate X chromosome and the mother will also donate X. That's 2 X's. So if one X is having issue, the other one will replace. But in a boy, X and Y, no replacement. If X is faulty, there is problem. So these kind of diseases are transferred by sex chromosomes, like hemophilia, blood. Some people, once they injure like this, blood will be rushing and it will be very difficult to stop it. Color blindness and so on. So more common in males that have one X. Females can be carriers, but they hardly fall victims. They hardly become infected or affected. So this is just an example. Please, you can go and study it. Thank you. By the time I share these slides, you can. If you have stayed to this point, yes, I salute you. You have tried. I understand. It is not easy to give time and attention to stuff like this. Meanwhile, you are doing yourself good though. So it requires training. It requires training and discipline for someone to sit and really learn. Some have dropped along the way. Some maybe even first 2 seconds they have gone. But by the way, I appreciate your patience for staying. Now, if you have not subscribed, if you have learned something at all, there is one thing I want you to do for me. 3 things in one thing. 1, like this video. Why should you like it? If you like it, YouTube will show to many other friends. And I have a goal. My goal is to reach many more serious students. I have a coaching group that I'm coaching students. They know. If you are part of them, you know. So by the time you like, YouTube will say, oh, people are liking this. Let me show it to many more. It will help me to reach out to more students and attract them, to assist them. I have a goal. Last JAMB, my aim was to have 250 students score 250 marks in JAMB. I could not achieve that, but I thank God that I've been able to still have students that score 300 plus, 250 and above, with a few having around 180. So this year, I want to raise the bar. Please, the only help you can do is to like, subscribe to my channel. YouTube will also say, oh, people are liking this, people are subscribing. Let me show it to more people. So that's the only help you help me do. Please, if you have any other thing you want to say, any other question, you can drop it in comment section. Don't go without liking this video. Don't go without subscribing if you have not subscribed. If you want to be part of my students, please also indicate interest so that I'll add you to WhatsApp group, provided you can be very serious. Thank you once again. Please expect the next video on the questions that I will be extracting from this heredity, variation, and evolution put together, and I will answer them very, very clearly. Watch out for that video, maybe tomorrow, in 2 days' time. I will drop it for you so that we'll be done with this topic. If you are my student, you know that we are taking topic by topic so far, and we are moving. If you are not, please, you can be part of us, provided you can be serious. That's my condition. All right, thank you once again. Please, I hope you have liked Okay, thank you very much. God bless you.